Hello, thank you for taking interest in our laboratory exercise. I'm Josh Titlow, introducing the lab on behalf of my co-authors Robin Cooper and Heidi Anderson. Since Thomas Hunt Morgan began isolating fruit fly mutations at the beginning of last century, Drosophila melanogaster has been an extremely powerful resource for understanding genetics, both in research laboratories and in the classroom. So optogenetics, or the use of light to activate photoreceptors in specific cells, is just as powerful for understanding how the nervous system works. The most important thing that I can say about this lab exercise is that it's fun and it works. Technically, it's very simple. Students measure how fast genetically modified fruit fly larvae crawl under a blue light and how fast they crawl when they are not in blue light. They quickly get the idea that light paralyzes the larvae, and they can collect data that clearly support this observation. So despite this simplicity, there's a lot of biology that can be learned about this preparation. It's ideal for teaching physiology and nervous system and striated muscles, of course. It gives students an active experience with genetically modified organisms and a research technique that is still very relevant. And at a basic level, students learn about invertebrate biology and life cycles. What we've presented is only the most straightforward experiment that you could use to quantify behavioral responses to optogenetic stimulation. There's dozens of fruit fly behaviors that could be controlled directly or indirectly using this simple setup. Since we finished this article, there's been a new channel rhodopsin that has been expressed in flies that is increased sensitivity and is more sensitive to red light. The advantage of this is that red light penetrates the cuticle of adults better, so you'd be able to use the same setup and do several different types of adult behaviors using the red LEDs and these crimson flies. So we'd encourage you to look into these flies if you're purchasing the setup for your lab. So my final point is that we're happy to discuss any questions or ideas that you have about the lab. Thanks for watching the video, and I hope you enjoyed the article.